Hello, everyone. I'm Chris Benko, and I'm pleased to welcome you to our webinar today on remote data collection and respiratory diseases. Thank you for joining us. I'm the CEO and one of the co-founders of Connexa Health, and I'm just going to take a couple moments to introduce today's session, and then I will hand it over to our phenomenal panel of presenters and speakers, including Dr. Singh, Ismailova, and Bhatia in just a moment. Just as a brief way of background on the first slide, I'll tell you a little bit about Connexa. I am one of the co-founders of the company, which launched in 2015 with support from the Merck Global Health Innovation Fund. Adam, if you could advance to the next slide. And actually, just before I go any further, just a reminder to anyone joining that this presentation is being recorded and will be archived for others to enjoy viewing. We'll post it probably within a few hours or no more than about a day or so after this recording. So Connexa is a company that works to develop and implement digital biomarkers that are designed to try and assess treatment effects using mechanisms that are typically remote at a patient's home, using wearable devices, mobile devices, apps, or sensors. And we do that primarily to support the development of novel medicines in the pharmaceutical industry. As a brief introduction to spirometry, I'm sure there are a lot of people who have joined this webinar because you're familiar with spirometry already, and you're curious about how it can be most effectively applied in a remote setting. I'm going to provide a quick introduction for those of you who might not be as familiar with spirometry. And certainly with COVID emerging, there are perhaps a lot more people interested in the impact that diseases can have on lung function beyond the respiratory diseases like asthma and COPD that many of us are familiar with from working throughout our careers. So spirometry is used to assess the health of the pulmonary system. It requires a person to breathe into some sort of airflow sensor that collects data about their pulmonary capacity. I had experience with pulmonary function tests as a child. I had suffered from asthma and allergies from my late childhood through my late teens. And so I recall regularly visiting Geisinger Medical Center in Danville, Pennsylvania, which is a phenomenal research facility, and being coached by a nurse to blow as hard as I could into a relatively large machine that took up what I recall being pretty much the area of a desk. That procedure can produce a lot of different values and data about lung function, more than 24. But two measures that are commonly used, particularly in drug development and in assessing the progression of disease, are FEV1 and forced vital capacity. FEV1 is the volume expressed in milliliters or liters, expressed by the lungs in the first second of what is typically a 6-second exhale. And forced vital capacity assesses the broader lung capacity in diseases like COPD. FEV1 is used frequently as a primary endpoint in clinical trials in asthma, particularly when trying to determine proof of concept. It's a very important indicator of someone's function and control when they have asthma as an underlying condition. There are important limitations to spirometry that lead us to the topic at hand today. Lung function is something that can vary quite a bit. It varies based on time of day. So your lung function in the morning and in the evening in most healthy adults and people with disease is going to change. It is also in many individuals with respiratory disease significantly influenced by seasonal or environmental factors. If you wake up with clogged sinuses or not feeling well due to environmental allergies or some other exposure, there's a very good chance that your lung function will not be as good if you were to go and perform a test that day as if you woke up with a very clear head. And that can be impacted by travel, regional variability, and so forth. It's also not something that's typically very convenient to collect on a highly regular basis. Patients need to go into a pulmonary clinic to perform in-clinic spirometry. And so it's not something you're going to get folks to do every day in the context of trying to get a granular understanding of disease. So what does this lead us to? Needing to control measurement variability is very, very important in spirometry. In clinical trial settings, even when we're assessing lung function in a clinic, there's concern about the consistency of the procedure, the way in which patients are coached, the way in which the machines are calibrated. all of those things can lead to measurement error that make it more difficult to understand the patient's underlying function. If you can get patients into clinic more frequently, more data is often better in terms of developing a better understanding of the patient's performance, but that's not always practical as part of a clinical trial. Patients don't want to be in the clinic or hospital more often than is necessary. And so this leads to the relatively large sample size of participants that are often required in drug development to achieve proof of concept or registration. Essentially, you need more patients to overcome some of the variability and noise associated with the data. Now, on the next slide, we'll talk a little bit about mobile or remote spirometry. This is not a new concept. It is something that people have tried to do for decades, dating back to machines that had to be plugged into old-fashioned modems to transmit data, or that recorded data in some form or fashion that patients could then bring into a clinic. But the idea is that you equip patients with a spirometer that they can take home and give them some form of instructions for how to perform a pulmonary function test by themselves. So this is without the coaching or supervision of the nurse or clinician that would typically be involved in clinic, but it provides the opportunity to collect readings that give you the much more frequent and granular understanding of what's going on with the patient's lung function. They might do it every couple of days. They could do it several times a day, depending on what we were trying to investigate. That data is then transmitted in some form or fashion, and those transmission modes have evolved over time back to the center or back to the researchers who are running the study. On the next slide, we will show you a modern way of doing this. So as opposed to older technologies that recorded things on some sort of disk or required plugging into a modem, these days, the evolution of Bluetooth and wireless technologies have provided significant advantage for how we can collect spirometry data. So this is a real picture of a Bluetooth ultrasonic spirometer that we've used in studies, including with Professor Singh, who's going to be speaking a little bit later. He recommended this spirometer to us as we evaluated multiple different technologies because it's ultrasonic. So it has no turbine, it has no mechanical working parts, which is ideal both in terms of the hygiene of the device, but also it is more sensitive to people that might have very low lung function and have more difficulty pushing through or forcing air past a fan. This device synchronizes with a mobile phone, and the mobile phone application is actually used in place of the nurse or clinician to coach the patient through the process of performing a lung function test. It provides verbal out loud cues as well as visual cues on the screen to let the patient know when they have exhaled the requisite amount that's required to provide the data for each maneuver. And 3 maneuvers, 3 full exhales, are typically required for a complete pulmonary function test. So what we do in our applications of remote spirometry is we deploy a proprietary application along with on a mobile phone, along with this Bluetooth spirometer device. And that allows us to synchronize the data from each maneuver into the phone and to test each maneuver for its full compliance with American Thoracic Society guidelines for that data. That data is then transmitted back to the researchers in near real time so we can see and assess, are we getting the data that we expect? And things like reminders and notifications can be used to help patients maintain a schedule that's consistent with the schedule of assessments for a study. So on the next slide, I would like to introduce today's speakers and panelists. First, we will have Dr. Dave Singh. Dave is a professor of clinical pharmacology and respiratory medicine at the University of Manchester. He's also medical director of the Medicines Evaluation Unit, which is an outstanding unit that we've had the pleasure of partnering with both on validation studies and in working clinical trials in patients with diseases such as asthma and COPD. We also have Conexa's Chief Scientific Officer, Dr. Elena Ismailova. Elena is a virologist by training who built her career at Millennium and Takeda, working primarily in oncology, biomarkers, and translational medicine before joining us 2 years ago. And she's also the co-founder of the Digital Medicine Society. And as our panel moderator, we also have our friend and former colleague, Dr. Gaurav Bhatia. Gaurav is currently the Chief Data Scientist at CareMore Health, which is a delivery system based in Southern California in the United States, but serves several million patients across the southern part of the US. He was previously a data scientist, a chief data scientist at Connexa Health, and remains an advisor to us. And his background is in molecular genetics and epidemiology. And so we're pleased to have him as one of our early collaborators on this work, rejoining us to take questions for our panel. So on my final slide, I'll just give you a brief outline of what our speakers are going to talk about today. Adam, if you could advance. Thank you. So we're going to talk first about spirometry and its use in respiratory diseases in more detail. What are the standard clinical practices for spirometry, as well as what are some of the implications around respiratory disease and COVID-19, which, as we know, has significant impact on lung function, and we're still learning and assessing what that means over time. We'll also talk about some of the challenges of developing drugs for respiratory diseases. And then specifically, what are the issues associated with moving clinical trial assessments to remote settings? As we set up front, there is a lot of potential value in being able to get more frequent data and reduce measurement error. With the pandemic, there is also the added consideration of quarantines and limiting patients' exposure to the clinic, particularly vulnerable populations. And then finally, we'll cover material that was recently published in Clinical and Translational Sciences. All of our speakers are co-authors to that paper, which looks at the comparison of in-clinic and at-home spirometry measures. So with that, I'd like to turn it over to Professor Singh to take us through the next section of the presentation. Dave?
Chris, thank you for the introduction, and thank you for setting the scene so nicely. If we could just go back one slide, please. So my job in the next few minutes is just to give you my view on how important spirometry is in different respiratory diseases. I'm going to focus on asthma and COPD, touch a little bit on COVID-19, but clearly spirometry has a great role to play across the breadth of respiratory diseases. And those of you who work in this zone will know exactly what I'm talking about. I've had the pleasure of collaborating with co-speakers, everyone else on this call at the moment, through the study you're going to see later on. So, and I'm sure you'll find that of great interest. But I'm going to cover in detail now two areas. What's the role of spirometry in clinical practice? And how has the COVID-19 pandemic affected that? Next slide, please. So here we have a list of different scenarios where we use spirometry. Diagnosis is probably one of the major areas. So particularly for obstructive lung diseases, then spirometry remains part of the diagnostic criteria. And I'll talk about COPD very shortly, which will bring that to life. But there are other situations where we commonly use spirometry, and I guess we don't always think about this. These are all listed out so that we can appreciate the breadth of how often spirometry is used. So for example, to assess pre-operative risk, to assess prognosis, I'm going to show you that with COPD. Screening situations, lung health, I'll show you that again. Now, once a patient has a disease, then monitoring over time becomes important. And for most lung diseases, where you either have an issue of reduced airflow or affected lung volumes, then spirometry is used over time, over years, to track disease progression, but also in real life to evaluate the effect of any interventions that clinicians may have, including pharmacological interventions. There's the whole area of disability or impairment evaluation, for example, patients taking part in rehabilitation programs, insurance evaluations, and legal cases. Then we have a whole load of other items where spirometry is used. So research and clinical trials, Chris has already talked about the importance in clinical trials. I'm heavily involved in clinical trials. I have staff performing multiple spirometry measurements every single day on patients across a breadth of respiratory diseases. So I won't read all of these out, but I think you get a flavor of the different scenario situations where spirometry is used. So moving on to the next slide. I'm going to talk now about COPD. It's a disease that I probably have the most interest in. And so I'm going to show you 3 areas where we use spirometry. Those of you who care for COPD patients will know exactly what I'm going to say next. So firstly, COPD can only be diagnosed with spirometry. So COPD is chronic obstructive pulmonary disease. The word obstructive gives you the clue you can only diagnose an individual with this condition once you've established that they have airflow obstruction. So a triad is needed. Patients have symptoms. If you then uncover that they have risk factors, commonly cigarette smoking, once you put those 2 things together, you confirm the diagnosis with spirometry. So this slide is from the GOLD initiative, and I'm part of the science committee of GOLD. We deal with a management strategy for practicing clinicians who care for COPD patients. And we put spirometry as the cornerstone of the diagnostic. Why? Because we thought this is relatively straightforward for clinicians to be able to perform across the world. There are debates around whether you should use other diagnostic techniques as well. For example, CT scanning can show you emphysema. But in the end, you need something that the clinician in any healthcare system will be able to implement, and we felt that spirometry was that measurement. So on the next slide, we see how spirometry actually is also a prognostic marker. So once we have the diagnosis, the actual numerical value that you obtain means something. So we split patients into 4 categories of COPD, GOLD 1, 2, 3, 4, according to their FEV1. So GOLD 1, they have mild COPD, so their FEV1 is 80 to 100%, and GOLD 3 or 4 is less than 50%, and we call those patients severe or very severe. And this is survival over years. It's obvious what's going on here. Lower FEV1, worse prognosis. So I'm showing you in the last 2 slides how we use spirometry to diagnose, how we use it to gauge what the prognosis is for an individual. And on the next slide, you'll see how our thinking has altered in the last few years to understand different types of COPD based on FEV1 telling us about lung growth. So this paper's in the New England Journal of Medicine. It combines 3 birth cohorts, patients tracked over decades. And the uniqueness of this is that these individuals had their FEV1 measurements available from early adult life. And you'll see there's 2 groups of individuals here, groups in red and green, who at around age 20 reach 100% predicted of FEV1. So this is normal lung growth during childhood and adolescence. Then there's a red and a black group who do not get to 100% predicted. So these people have suboptimal lung growth. So I'm just stating the obvious, what you can see there. Why do these people have this? Variety of factors. So poor nutrition, childhood illness, perhaps environmental, so exposure to pollution, secondhand cigarette exposure, premature birth. So we find small lung volumes at birth tracked throughout life in some individuals. And maybe it's a combination of these factors. Now, the relevance of this then is you see the black and the red individuals are people who then smoke and go on to develop COPD. The black individuals, the line, if you look at the slope of it, it's actually relatively shallow. And they have 2 reasons for developing COPD. 1, they started at a low volume, and 2, they smoked. but it's a different trajectory of COPD. So this gets you into thinking about the value of spirometry to track individuals' lung health throughout life and discovering individuals who in early life have suboptimal lung function, making public health interventions, such as not pharmacological ones, such as education around stopping smoking and lung health to try and stop them following that black line and keep them on the yellow line where they won't develop COPD. So let's move on and look at the next slide, where we'll think about some of the challenges we now face across respiratory function testing with the COVID-19 pandemic. So we hear a lot on the media about the transmission of this virus. Is it aerolized? Is it transmitted through surfaces? And the answer is both, but what we don't really understand is the degree to which it's either, is it 90% aerolized and 10% surfaces, or the other way around? And we could debate that for a while. But what's clear is it's incumbent upon us to minimize risk, because we do know that the virus can be aerolized. And so procedures that may generate an aerosol that could increase transmission risk, we have to be careful. And spirometry potentially is one of those. So here are the American Thoracic Society recommendations, and that they quite rightly say that we have to put measures in place to protect staff and those individuals being tested. So one of those measures, for example, is rapid COVID-19 testing. Can you get a quick turnaround of the test, say the individual is clear, and then you can do spirometry. There are other cleaning measures that we put in place, and social distancing measures, etc. But ultimately, spirometry, even during this pandemic, remains a test that you need to diagnose some conditions, for example, COPD. And in our laboratory, in our hospital, with these appropriate measures, we are carrying out spirometry at a lower volume because of social distancing, but we're carrying it out when necessary, when it affects your decision whether to treat a patient. So on the next slide, you'll see another aspect that's growing and is linked to COVID-19. And some people call this the long COVID-19. It's patients who are suffering with persistent symptoms despite recovering from the acute phase. The pathology is complex. You all know that there's an acute lung injury in a subgroup of individuals who then are hospitalized. They become hypoxic. They have a mixture of hyperinflammation, increased coagulability. They seem to have a sort of vasoconstriction in the lungs that contributes to a hypoxia, in addition to many of them having emboli. So this multi-pathology actually appears to lead to alveolar destruction. And although it's been called ARDS, the pattern in the early stages is not typical of ARDS because the lungs are extremely compliant. So the lungs are not stuffed full of exudate, which causes decreased compliance. They're relatively compliant. The problem is, because of the alveolar destruction, you can't ventilate them properly. Now, this seems to take a while for the lungs to recover from. And these individuals now are going to need care, and they're going to need follow-up. They're going to need follow-up with lung function testing, including spirometry. Some of them will need CT scan follow-up over time. And this is something that we need to put in place. So on the next slide, I just wanted to outline, and Chris has really done a great job of dealing with the details of this, but I wanted to outline how we're moving forward in this area. So I have a clinical research unit where we use predominantly laboratory-based spirometry because we have patients coming into the clinic, we have patients staying for a length of time, but often our patients on clinical trials, that's why they stay here, they might stay for 12 hours to have their lung function measured after taking a dose of a novel medication. But often they'll be on long-term studies and they'll have to come back at intervals. And one of the things I've been discussing with Elena and Chris and Gaurav over the last few years is the applicability of portable spirometers, making life easier for patients, the advances in technology, making these work through smartphones has been fantastic. And that really leads us on to what you're going to see in the remainder of the presentation. So as we move to the next slide. So I just want to show you now some of the existing data on mobile spirometry. And I've picked, there aren't many studies that have actually published in this zone. And I've picked on this one, which is an asthma study. And it's a very large study where different doses of inhaled corticosteroids were evaluated by clinic spirometry at intervals or daily spirometry. And one of the beauties of daily spirometry, to state the obvious, is that it's daily. You can get frequent measurements, whereas clinic spirometry has to be done at intervals because patients practically can't come back. So here you see 3 different doses of inhaled corticosteroids compared to placebo and for the home-based measurements. And this is the type of result you would expect because actually it's very hard to evaluate a dose response effect of inhaled corticosteroids in clinical trials. But the next slide is probably the most interesting one. It's the correlation between the clinic and the home spirometry measurements. And this gives you a very good feel that there is a good association between those measurements. And this is the background data. This is a multi-center study. The data from here isn't in the paper, isn't reported in any greater granularity or detail, but we felt we wanted to look at this in greater detail and get more of a handle on the statistical power afforded by home-based measurements with more frequent measurements compared to the less frequent clinic measurements. So with that, I'm now going to hand on to Elena to take us through to the rest of the presentation.
Thank you very much, Professor Singh, for giving a very comprehensive overview of spirometry applications in clinical practice. My part of the presentation will be focused predominantly on application in clinical trials. As you all know, that drug development in respiratory diseases presents formidable challenges. And one of the top challenges is disease heterogeneity. The condition that we called asthma is actually a constellation of different diseases that may or may not share the same symptomatic features, underlying mechanisms, and treatment response. For instance, childhood asthma is a very different disease than adult asthma refractory to corticosteroid. And in order to develop an experimental medicine, the efficacy needs to be assessed across population in pediatric, adolescent, and adult patients. And the response may also vary depending on the disease stage. A cross-industry group got together to analyze challenges of drug development in immunologic diseases, including asthma, and they provided some recommendations of how these challenges can be addressed. They analyzed the attrition rate for drug candidates between different stages of drug development. The highest attrition rate was observed between phase 2 and phase 3 studies. One of the underlying reasons for such a high attrition rate is disease heterogeneity. And the culprit was pointed to the phase 2 studies not being adequately powered and not representing the patient population in phase 3 studies. And the group has suggested to increase the sample size in phase 2 studies. Today, we offer you an alternative solution to increasing sample size in clinical trials. And as you may have guessed, this solution includes mobile spirometry measures. In the next slide, I'm going to share with you the results of our recently published paper in Clinical and Translational Sciences, where we answered a question how mobile and clinic spirometry measures compare and how they can be used in clinical trials. The next slide, please. This study was done in collaboration between the University of Manchester, Regeneron Pharmaceuticals, and Connexa. And key questions we tried to answer in this study are related to comparability of mobile and clinic spirometry measures. Are these measures equivalent? Are they comparable? Can they be combined and used interchangeably? When patient comes to see their doctor and visit a spirometry lab in the clinic, compliance is not an issue. They are supervised during the procedure by a nurse who provides guidance and coaching. But will people contribute and do the same thing while they are away from the hospital and unsupervised? As you learned from the previous part of presentation, it requires an effort on behalf of the patients to contribute spirometry data. Will patients put forward their best effort when they are away from the hospital? So in the next slide, I'm going to share with you the outline of the study. And during this study, the patient with moderate asthma were asked to come to the clinic weekly, contribute their spirometry data, and they were also given a handheld spirometer, trained to use this spirometer, and before they were heading home, they had to demonstrate at least 2 successful maneuvers in producing the spirometry data. At home, they were asked to contribute spirometry data twice a day, in the morning between 6 and 10 a.m. and in the evening between 6 and 10 p.m. They also were offered a third option to contribute the data, which was not absolutely required, in the middle of the day. Each time they had to do 3 expiratory maneuvers, and the highest value was taken for the analysis. The analysis of the data included the totality of home and clinic spirometry data. They were compared using various statistical tests, and we also did a power simulation exercise to see how both measures are compared when we asked a question about powering a certain study, expecting an outcome of a certain effect size. In the next slide, I'm start to sharing some of the data. So these were moderate asthmatics. 12 people consented to participate in the study. They had a median age of 41 years. They were predominantly men, and they met study inclusion criteria such as pre-bronchodilator FEV1 values, absolute and predicted. Patient demonstrated bronchodilator reversibility, a hallmark of asthma. They were non-smokers and were using standard of care medication, inhaled corticosteroids, and long-acting beta agonist. First look at the data confirmed that our concern about patient data contribution was addressed. Once a day data compliance contribution was 85%, which is really good, and twice-a-day data contribution was 70%. These results are comparable to another study in COPD with a longer duration of 52 weeks, implying that maintaining this level of compliance for longer periods of time may be feasible. The next slide, please. As Chris already explained, asthma has diurnal variation. Changes in FEV1 values depending on the time of the day vary even in healthy individuals, and differences in FEV1 values between different times of the day are bigger in patients with disease. The FEV1 values are the lowest in early morning hours when disease is at its nadir. When we looked for distribution of FEV1 values in our study, we had to separate them depending on the location where they were taken and depending on the time of the day. For this analysis, we used the residuals of the mixed model, and the residual shows you the deviation from the average value for a particular patient. We had to use linear mixed models because we had a bigger number of mobile spirometry values, and we had to adjust for subjects' averages. Our data had shown that the morning mobile spirometry measures were lower, and the difference was statistically significant compared to the evening mobile spirometry values. The morning values, if we compare measures taken in the clinic and remotely, the remote measures were lower, but not statistically significantly different. There was no numerical difference in values between morning and afternoon measures in the clinic, and the difference between evening and afternoon measures taken in the clinic were not statistically significant. The pattern we are seeing here is consistent with asthma diurnal variation. From here, as shown at the next slide, we proceeded to comparing directly clinic and mobile spirometry measures, and because of disease diurnal variation, we had to separate them between morning and afternoon measures. The correlation for both categories of values was very strong and showed excellent correlation. When we looked at the limits of agreement to see if there are any biases and how these values compare to another, we have noticed that the mean differences between morning morning spirometry and clinic measures were bigger than the same result for the evening and afternoon values, again, being consistent with asthma diurnal variation. We also asked a question how these measures compare for test-to-test repeatability, and we used interclass coefficient test to see how consistent are the values across the subjects and how well they discriminate between the subjects. The interclass coefficients were very comparable between clinic and mobile measures, indicating high repeatability in both cases. And at the next slide, I'm going to share with you probably the most interesting part of this research when we asked the question about power of both measures to predict the outcome of the treatment. We performed a power simulations using both measures, and this is an exercise that a study statistician would do to answer the questions: how many subjects should be enrolled in the study if we want to see certain difference between control and treatment arms. The question we're asking here is slightly different. We ask it a question: if we take FEV1 data from all study subjects and we want to detect the difference of 0.2 liters in FEV1 values, what would be the probability that we will be able to see it with clinic or mobile spirometry measures? So what you're seeing in this graph is 4 simulation scenarios. The first one was using only first and last clinic spirometry data, and this is how usually the outcomes in clinical trials are being computed. And this scenario yielded the result of 35% probability of seeing the effect in FEV1 of 0.2 liters. When we included the data shown in green here from all clinic visits, we saw the power of 42%. The next blue line shows the results of simulation using once a day spirometry, mobile spirometry measures taken in the morning, and the power jumped to 75%. And when we used all available mobile spirometry data shown here in magenta, the power was 95%. The simulations for 4 scenarios were done similarly. The main difference here that we adjusted the model for the time of the day for mobile spirometry measures because we had increased degrees of power and we could afford statistically this adjustment. This is an example of how mobile spirometry measures can help reducing the sample size. And also, if you think of the scenario shown here in red, how it can help eliminating the effect of random anomalous test result. Chris gave you an excellent example in the introduction of an asthma patients having a bad day. And increasing degrees of freedom give us a chance to correct for factors that inherently can affect variability such as time of the day or season or some other environmental exposure. In my next slide, I would like to summarize the result of our study and say that we have here an opportunity to figure out how to reduce the sample size in clinical trials with FEV1. And this tool can be used to study disease in subpopulation addressing the issue of disease heterogeneity. I also would like to highlight that this was a single center relatively small size study, and the results should be interpreted with caution because generalizability of small studies needs to be confirmed in larger studies, and this is one of our future directions. We would like to test this hypothesis again in a larger study with known treatment controls, such as drugs approved for treatment in asthma and COPD, montelukast or tiotropium would be good examples. We also want to assess the impact on sample size in a broader disease population going beyond moderate asthma, looking for other disease severity grade of asthma or in COPD, and hopefully in a longer duration study. I'm going to stop here and open this conversation to Q&A. Thank you.
Thank you so much, Elena and Dave. So please feel free to submit your questions through the Q&A in Zoom. We wanted to make sure that we allowed time for our panel to address questions that you have about today's presentation. And we have Gaurav joining us for that discussion, particularly for those of you that have questions around some of the statistics and methods. I have an initial question that Professor Singh looks kind of geared for you. Are there specific clinical trial scenarios that you think would be especially well suited to mobile spirometry based on the types of studies that you're seeing come through your pipeline at the Medicines Evaluation Unit?
Yeah, thanks for the question. So I think, Chris, you mentioned it earlier in your introduction. No, it was I think Elena mentioned it as well around phase 2 trials. So that's one of the big areas here where this technology would be useful, because phase 2 studies, for safety reasons, you often have limited numbers of patients. Then if you were to be able to, for example, have an asthma study where you had a novel anti-inflammatory drug and you only wanted to expose a limited number of people to this, so your study is in the tens rather than the hundreds. It's a 4-week study. That's the sort of study I would be looking at. So we often see those studies done with large numbers, with FEV1 being done every week. So you've exposed huge numbers of patients and you probably don't have enough power. Well, these dense measurements, we may have thrown some good drugs away in the past because the studies with clinic spirometry were just not powerful enough. So that would be what I've learned from this pilot study.
Thank you. That's incredibly helpful. Gaurav, could you speak a little bit to the notion of whether or not these techniques might be interesting in terms of modeling homoscedasticity or heteroscedasticity of treatment response?
Sure, yeah, it's a good question. It may get into a little esoterica, but effectively, if you think about the standard assumption that underlies the sample sizing calculation that goes into any clinical study. So this is, your kind of vanilla, what would be the power of my a phase 2 or phase 3 efficacy study if I were to do a t-test between cases and controls. The fundamental assumption is that the effect size is a constant. It's one number, as opposed to being drawn from a distribution. And that in and of itself, so when we think of variability of response, this is going to be a significant source of loss of power for most studies. In the context of mobile spirometry, I guess I'd say at its simplest, it all sort of comes down to power. And the fact of having many measurements on a per patient basis allows you not only to overcome the problem of variability in response because you have so many more measures, but also allows you to assess the existence of the problem. So one of the fundamental challenges in kind of your traditional study is that you really don't have the ability, in kind of the most fundamental sense, you'll have a very hard time assessing whether or not different patients are responding differently to the treatment. Whereas in a statistical sense, it's actually quite straightforward to take multiple spirometry measures and ask whether or not there is individual level variability in response. And you can simply estimate that as a parameter, provided you have enough data points, which mobile spirometry enables you to do.
Thanks, Gaurav. That's super helpful. And you're great at simplifying or making those concepts a little bit more straightforward for those of us without the stats background. Dave, I think this next question is probably good for you. I know in the context of our study together that we did not assess this, but you may have some additional experience to share. Have we looked at the differences in variability of measurements that are performed at home with no coaching versus measurements performed at home with some form of coaching done by telemedicine or remote interaction with a technician? So not something that we did in this study, but do you have any other experience to share in that regard?
I don't actually, and it's a good question. But so I think our study was quite helpful in reassuring that without that personal coaching, we still had a good result. One thing that was interesting about our population, you may have noticed when Elena presented it, the high degree of reversibility over 500 mils. Now, why is this? This is a bit beyond normal variation, normal reversibility. And it's because of what we all know about most asthma patients. They don't take their medicines regularly. So when you force them to come into the clinic and you say, take this Ventolin now, they improve by 500 mils on average. So we actually had a suboptimal compliance population who then, with mobile spirometry, with the prodding of our staff, because our staff, not coaching, would remind them they're on the study and they come back in every week, so they weren't just left alone for weeks and weeks. But this population who were suboptimally adherent with their medicine gave us a good result for adherence to their spirometry in a clinical trial setting. I found that interesting.
That is really fascinating and relatable to those of us familiar with the field and the treatment patterns. Elena, I think this next question is probably best for you. Could you speak to the status of acceptance of mobile spirometry data by health authorities and regulators, especially in the context of phase 2 or phase 3 trials of new investigational medicines? Of course, phase 2 and phase 3 can be very different in that context, but please, I'd love if you could extrapolate there.
So I'm not aware of any approved drugs on the basis of mobile spirometry. If you are considering mobile spirometry for your clinical trial, I would encourage you to reach out to appropriate regulatory authorities and discuss with them as early as possible. Again, my prediction about what would be required for this conversation is to show the equivalence of clinic and spirometry measures in specific method and device that is being deployed in your study. And today we gave you an example of how these equivalence can be established. I know both European and US regulators are very interested in using remote measures, including mobile spirometry, and would probably welcome technological innovations making clinical trials more efficient. At least this was our experience in interacting with regulatory authorities.
Elena, I wholeheartedly agree. And one thing I will just add from experience that you've heard me say before, having worked in the pharmaceutical context my entire life, the decisions being made around investments between phase 2 and phase 3 inside a pharma company are enormous. And so their resources to evaluate these decisions are a big deal. And sometimes their standards may be different and may ask for even more data and more rigor than a regulator might, because of the gravity of how significant it is to do a phase 3 program in these areas. Dave, this one I think would be great for you. We're not the experts in this at all. Can mobile spirometry be used for a confirmatory diagnosis for COPD or asthma?
So it's not used in that context, but I think it can be used. So as we get more confidence with the devices, as in the one we presented today, particularly in this COVID pandemic, you explained, Chris, that we get feedback according to the ATS criteria. So we will know whether that is a good standard spirometric maneuver. The only thing you don't know really is whether the individual themselves did it or whether they handed it to someone else. But for the actual quality of the maneuver, we can be confident in it. So I think it's a practical thing to think about when I talked earlier about aerolization, etc. That one in the current pandemic, you could actually think about doing this in, the healthcare staff are not near the individual for infection control reasons.
It's amazing how much the necessity forced by this situation may drive us to innovate beyond what we would have done otherwise. It's a feature we've seen in other areas of the pandemic. Well, I want to thank all of our speakers and Gaurav for joining. I'm excited to, even though I was involved in this study and I've seen the paper, I learned a lot myself just hearing you all go through, particularly some of the emerging and evolving issues as we're seeing in the current landscape. So this was really fantastic. I appreciate your time. Panel participants, thank you for joining and for your great questions. And for those of you that have colleagues that would be interested in this content, as I mentioned up front, this is recorded and we will be reposting it shortly. So thank you all and have a great rest of your day.